From transcription, we obtain a strand of mRNA, or messenger RNA, which pairs to the template strand of DNA, with the exception of thymine being replaced by uracil, as RNA does not contain thymine. After we obtain our mRNA strand, the strand is taken out of the nucleus into the cytoplasm to go through the process of translation. In this process, mRNA enters a ribosome through the A site, or entrance site, starting with the 5' end of the mRNA. Now, mature clover-leaf-shaped tRNA molecules are brought in to match the nucleic acids on the mRNA with base pairing anticodons. Here is where we are brought into Francis Crick's adapter hypothesis. Crick proposed that there must be small RNA molecules that can receive and give specific amino acid sequences that are determined by what we call triplet anticodons, or 3 matching nucleic acids. Crick thought that these molecules, as he figured out to be secondary structure tRNA molecules, had to pair exactly with the nucleic acids coming through the ribosome, and that would code for specific amino acids to chemically combine through hydrogen bonding to tRNA's anticodon template. Before Crick determined that tRNA was responsible for carrying the anticodon template and binding to amino acids, his partner Sidney Brenner suggested that there were 20 different kinds of adapters that carried anticodon templates, and each adapter was specifically made to bind with specific amino acids. Now, looking at the chart, you can see the mRNA entering the ribosome at the A site, starting with the 5' end. As it enters, adapter molecules, or tRNA molecules, carry pairing anticodon triplets. They are adapted specifically to match these sequences in order to code for amino acids. At the P site of the ribosome, processing occurs, where amino acids begin to bind to the tRNA molecule. We can determine the coded amino acid from the amino acid dictionary shown here. Note that some anticodons code for a start and stop amino acid so that the chain can start and end. Now, once the amino acid is bound to the adapter tRNA molecule, the tRNA and amino acid moves to the E site, or where they exit the ribosome. Here, the amino acid binds to the previous amino acid and a polypeptide chain is formed, and it is called this because peptide bonds are what bind the amino acids. From here, the tRNA molecule detaches from the amino acid. This ends translation and begins the translocation process of the polypeptide sequence. Alright, that's adapter hypothesis in short. Thanks for watching, and this video was brought to you by the following sources.